see if I can escape out of here. Okay. So, nutrigenomics. What we eat influences our gene expression. And one of the interesting things about the agouti is, what did they feed them so that they had normal offspring?
B12 and something else.
Vitamin B12, which is folic acid.
But they said something else, and I didn't catch it because I was writing down the B12.
B12. They said something other than folic acid?
Yeah, they said a different vitamin as well.
Oh, I only caught the vitamin B12 and folic acid. Folate, maybe they said?
Maybe.
Okay. So one of the really interesting things about this is, why do we give this, or why do we have females who are thinking about becoming pregnant take prenatal vitamins? For the folic acid, right? And why? What effect does a lack of folic acid seem to have? So what are you more likely to get in your babies? Does anybody know that?
Yes.
It's affects the development of the spinal cord.
Spinal bifida.
Yes. So lack of folic acid in humans during pregnancy causes spina bifida. Okay, so that's where the spinal cord is actually outside of the vertebral column, and it's usually like a sac of nervous tissue. Right? So that's why they have females take one of the reasons why they have females take prenatal vitamins, so they have all the necessary nutrients so that the developing embryo and the developing fetus turns out normal. Okay. So if we look at, okay, so if the agouti gene, right, if that gene is on, the individuals were prone to cancer and diabetes. And so they fed them that, then they were able to produce the methyl groups, the folic acid, allowed them to be able to produce the methyl groups so that it would attach to the DNA and turn those genes off. And so they were able to have offspring that were thin and healthy. So this is really interesting because if you hear talk about our problems with obesity and other diseases, is it seems that today it is much, much harder to be a lower weight than it was in the past. So if you take people that like in the 1980s and you look at what they ate and you look at what their exercise they did, they were thin compared to us. So they say that we are about 10% heavier today than we were in the 1980s with the exact same diet and the exact same exercise. And so there might be a truth that there might be something that we're taking in, be it, synthetic chemicals that are found in food production, or something that we're not taking in that is affecting our gene regulation so that we tend to be heavier today than we were in the past. Does that make sense? Okay. Okay.
So the number 7 is like how they were able to turn off. That's just they
fed them folic acid or vitamin B12. Yeah. So number 7 or 6. That was 6. So 7, early on, when the twins are young, their epigenome is very similar, but as they age, they experience different environments. So if you think about maybe a twin would smoke, one smokes and one doesn't, their epigenome is going to be dramatically different. And drinking and doing drugs, it used to be that we thought that drinking was only a problem if you were pregnant, right? But drinking alcoholism might actually affect the epigenome. And might actually be passed on to the offspring. So another really good example of nutrigenomics, let me show you this one, is in the bees' diet. And so when you look at the difference between a queen bee and a worker bee, all it has to do is with what they have been fed. And so in order to produce a queen bee, the worker bees produce a substance that is in royal jelly. So they call it royal jelly. So they feed only one larvae the royal jelly, and it silences a key gene, right, which codes for an enzyme that causes genome-wide silencing. And so the genes, the queen genes are epigenetically silenced. And if they're not silenced, then the larvae will form into a worker. Okay? So the difference between queen bees and worker bees all has to do with what they're fed during embryonic development. Okay, so the other example is on the back of that handout. Did we answer all the questions first? Let me get it. So number 7 is as they age, there's an increase in epigenetic change because they experience different things in their environment. And then nurturing, the effect is that a mother who is not nurtured could pass on her epigenome to her offspring so that they are also tend to be high stressed. And then number 9 was that you can give them drugs that either add tags or remove tags, and that can influence gene expression. So this is a big deal right now because we thought that when we passed our genes on to the next generation, all we were passing on was our genes, not what we have experienced in our life. And so the one example that I think is really interesting is this one where fear, so this is on the back, so fear can actually pass from one generation to the next in mice. So for example, if you train mice to be fearful of the smell of cherries, right, cherry blossom smell, then their offspring will also fear that odor, even though they have never experienced it before. And so sometimes that is referred to as, we call this, in sociology, you'll see it a lot, they call this intergenerational trauma. So the traumatic events that happened in the parents can influence the epigenome and actually be passed down into the next generation. So, you know, if you think about males going off to war and then coming back with PTSD and then having children, that experience of being in war could have influenced their epigenome, and they could pass on some of that fear to their offspring. Okay. Any questions about that idea? Okay, so besides just simply looking at the epigenome, we do have the ability to have switches. And actually, we talked about switches when we were talking about the switching on and off of the gene that codes for lactase. And so we can talk about the switches, which are called operons. So if we have, let me see if I can switch this to, oops. Just switch this to only, okay. So if we look at a piece of DNA, they call this upstream. I'm going to put 3 different sequences here on my, this is just like a piece of DNA, a diagram of a gene. This is what is referred to as the promoter. And this is where the RNA polymerase binds. And this is where transcription can occur. So RNA polymerase binds in preparation for transcription. Okay. This second region is called the operator. And then this is what is called the functional gene. So sometimes they talk about the promoter and the operator being upstream from the functional gene. So these sequences of nucleotides, they're just like the ATCG, right? But they're not, they don't code for anything. They just allow, they kind of tell the body that this is where a gene is located, and you can start transcribing it. And then the operator is really important. That is actually the switch. So we have molecules called repressors. So the repressor molecule can bind to the operator and turn the gene off. Okay. Now this is what happens in very simple organisms like bacteria. And so when we talk about these gene regulation models, we're looking at really simple prokaryotic organisms like bacteria. It's a little bit more complicated in eukaryotic organisms, which is kind of beyond our scope of what we talk about in a 100 level class. And you could learn more about it if you're going to take 300 level genetics. Okay. So the examples of these models, there's two that you need to know. The first one is called TRP, TRP, the TRP operon. The TRP operon regulates the production of tryptophan in bacteria. So tryptophan is an amino acid. This is what is referred to as a repressor operon. Okay, and so when we look at the repressor, let me see if I can find my notes. We say that it is turned on, so gene is turned on unless tryptophan is not present. So you can get tryptophan, and bacteria can get tryptophan from eating other things. So if the tryptophan is not present, or if the tryptophan is present, the gene is turned off. Okay. So in your book, there is a diagram of this. And it looks something like this. Okay. So this is my producer, my operator, and notice that the repressor, if tryptophan is not present, the repressor does not bind to the operator. If tryptophan is present, it will bind to the operator and turn it off. And so that regulates, that means that if the bacteria only produces tryptophan if it needs it. Okay? So we would say that tryptophan, the presence of tryptophan represses the gene and turns it off. Okay. So the other example is what is called the lac operon. So lac stands for lactase. Okay. And this is what is referred to as an inducer operon. So this induces it. Okay. So the gene is turned off unless lactose is present.
Michelle?
Yep.
I'm sorry. When you flipped back and forth, I got confused.
Okay.
When back at where you said repressor.
Okay. So this is a repressor. The tryptophan is a repressor because
In the notes you said repressor, and then you put gene is turned on unless tryptophan is not present or present.
Right?
the gene is turned on unless tryptophan is not present.
That's not. Okay.
Not.
Sorry, just shifted real, yeah, thank you.
But then you said the presence of tryptophan turns the gene on.
Off. Right? The presence of tryptophan will turn the gene off because the gene is always turned on unless tryptophan is present. So tryptophan actually represses.
That's right, so gene is turned on unless tryptophan
Is
present.
not present. Presence of tryptophan will turn it off.
Off.
Will turn it off.
presence of tryptophan turns it off. So tryptophan is a repressor. It turns the gene off, okay? So in this case, the gene is turned off. That's its default. It's not on like the other one. So it's turned off unless lactose is present, okay? So lactose causes.
I still think it should say gene is turned on unless tryptophan is present.
Yes.
Tryptophan makes it turn off.
Yes,
Yeah,
gene is
correct.
turned on, sorry. Gene is turned on.
So take out the word not.
It's out.
Okay. Okay, sorry about that.
Okay,
That's what-
really am feeling.
Lactose causes the repressor to unbind.
Is this another repressor?
This is an inducer. So the lac operon. So what this means is lactose turns the gene on rather than the presence of lactose turns the gene on rather than off. So that's why it's called an inducer. Okay.
That's confusing.
Don't flip that screen.
We're all with you.
This is in your book too, if you want to review that. Okay. So what this does is you don't want to produce an enzyme if the enzyme, it doesn't have a function, right? So if you're a bacteria, if you had lactase and you were producing it and there was no lactose in the environment, then it would be a waste of energy and building blocks, right? And so this gene is only turned on if that food is present. And so that's the idea of the lac operon. Okay. So can I flip it now?
Yes.
Okay. So notice that the repressor is on the operator and transcription does not occur. But if lactose is present, it binds to the repressor. The repressor unbinds from the operator, and then transcription can occur, and then subsequent translation of the messenger RNA into protein would occur in the cytoplasm. So this is what the simplest form of gene regulation looks like. In eukaryotic cells, like we are eukaryotic cells, it is not this simple. And so in eukaryotic cells, you have many different regions upstream from the DNA. So notice this is my gene, but this is all the upstream part of it. And it actually has to create a loop. And then you have parts of them kind of binding to other transcription factors and proteins and activators, right? And so this just, I'm just going to tell you that the eukaryotic gene regulation is much more complicated than prokaryotic. But it still is kind of the same idea, is that you have switches that can either turn the gene on or turn the gene off. Okay. So in this particular instance, this just shows, oh, this is just unlabeled. So this is the promoter region, and then the subsequent gene. Okay. So we are going to go to the very last chapter, and it is on biotechnology. So when we talk about biotechnology, one thing that you've probably heard of is what is referred to as the Human Genome Project.
So when was that video made? Because they said there's a 10-year, it was a 10-year.
Yeah.
I'm curious when that.
You know, I don't have my glasses, so no, of course I can't see that fine print. But I'll bring the video to you.
Is that done, that study done?
I don't know. That'd be interesting to find out. There's lots of stuff in the media. If you follow science magazines and blogs and stuff, the epigenome is huge studying it. So I don't know. Okay, so the Human Genome Project, when we look at, this was between 1990 and 2003. So essentially we have mapped the human genome. And what this means is that we took kind of a typical human, and we tried to find out what the sequence of A, T, Gs, and Cs were. So when we look at the sequence of nucleotides, in a typical human, okay, if we look at the size of the genome, which is all the DNA, it is 3.7 billion bases. Oh no, sorry, 3.2. 3.2 billion base pairs. So remember that the DNA is double-stranded, and so A pairs with T, G pairs with C, and so this is the size of our human genome. Now, if you look at what the human genome does, we have probably over 200,000 proteins in the body. So when we look at the proteins, we have, this is what is referred to as the human proteome. So think of the genome as the sequence of nucleotides. The proteome would be all the proteins that are produced by your body, right? So there's lots of proteins. And so when they were sequencing the human genome, they thought that they would find that there were a lot of genes. But the big surprise came when they sequenced it and they found that there were not very many genes. And so there's about 21,000 genes. And so this was a huge surprise. Right? That we do not have very many genes. And so one question is, how do those, if we have 21,000 genes but over 200,000 proteins, how do those genes code for the proteins? All of those proteins. And I gave you one clue, which remember we talked about exon shuffling, right? So remember that the DNA can be transcribed into messenger RNA. So exon shuffling explains how a single gene could code for multiple proteins. Okay, so now we have to figure out, you know, how this works, how we have so few genes, but so many proteins in our body. So if we look at DNA testing, since the human genome, they have what is called direct-to-consumer testing. And some of you, I'm sure, have heard of this. Right? So instead of going to a doctor and saying, hey, I would like you to test my genome and tell me what genes I have and if I'm at risk, if I'm a carrier or at risk of having a disease, instead of going through a doctor, you can now just simply get a kit. You can pay for it, and they will send you a kit, and one of them has you spit into a test tube, and then you send your spit back, and they look at your DNA, and they look for specific variations in the DNA. So one example of a company that does this is called 23andme.com.
I actually just got my results back
Oh, cool.
this week.
Yes.
With my mom. It's pretty interesting.
Yeah. So this is the direct-to-consumer testing, and you might have heard about these guys because the FDA a few years ago shut them down and said you could not tell people about any health-related genetic variation. You can only tell them about your ancestry and your typical traits. And the concern is that if people found out that they were predisposed to cancer or predisposed to diabetes, that they might take the situation into their own hands and they wouldn't go see the doctor and the doctor wouldn't help them, right? And so there was this idea that the consumers should have limited knowledge of the data, right? And they just won and maybe the opposite would be true. Like if it told you you weren't prone to diabetes, but your doctor said you were, you needed to lose weight, maybe you would make changes to your lifestyle that would not be good, right? So the FDA just kind of opens that back up so that not only are they giving you ancestry stuff, but they're giving you limited, they have more information than they hand out, limited ideas about your health.
Okay. And they make you sign
Yes.
a bunch of disclosures.
Yeah, and they also, the concern here is that what are they doing with the data? And why would they do this? And it's really cheap, it's only like $50 or something like that. And what they're doing is they're creating a database. And this is what is referred to as the genome-wide association studies. So they're not really interested in you as an individual and your health. What they're interested in is gathering data. And so, for example, if you had this population that was diabetic, right, and this population that was not diabetic, and you did you had like 100,000 people here and 100,000 people here, and you looked at their DNA and you saw where their DNA was similar. So if all the diabetic individuals have in one part of their genome the same sequence, and all these guys don't have the same sequence, then maybe that is where they need to look in your genome to understand diabetes. Does that make sense? Okay. So what they're hoping is that they're going to create these huge databases, and that you are going to, if you get this done, they're always trying to have you take little, not quizzes, but little surveys. So they need the surveys, right? They need to know, we said that you were going to be overweight. Are you overweight? What do you weigh? Because that is the information that they actually need. And so they want you to do all these surveys so that then they can start teasing apart. And then perhaps they can sell this information to a biotechnology company. The biotechnology company is looking for drugs to treat diabetes or cures for diabetes. And so they might be able to pay for this, give them money for the research that they have done. Okay. So that's kind of the idea of the 23andMe program and others that are similar to that. So if we look at the 23andMe and what it kind of tells you is that it doesn't, in many cases, look for single genes, but it again, is just looking for that variation that might be significant. I guess they're having a sale. Did I have it? I thought I logged into my account here. I don't think I did. Oh, here we go. Okay. So some of the things, oops, signed me out.
And Michelle, are you the only one in your family that did it, or did you have other blood-related people do it?
I am the only one in my family that did it.
We just did it. I did it, and then my mother is doing it as well. And so
that would be interesting. My family is phobic of science, and so I actually did it because I was interested in my my mom died of Alzheimer's and my grandmother died of Alzheimer's, right? So I was really interested. And in the past, they actually did Alzheimer's. They don't do that now. I don't think they test for that. Did you get a feedback about Alzheimer's?
It said dementia and Parkinson's, but it didn't specifically say Alzheimer's.
Yeah. So there is an allele, there is a variation that is linked to Alzheimer's, and I have one copy of the bad allele and one copy of the good allele. So I was really happy about that. But I tell you, I presented that to my sisters, and they were pissed off. They're like, and they're like, why would you even want to know? I don't want to know. And my sister even said, "You're gonna die before me anyway."
Okay.
So yeah. Family fight over 23andMe. I do not want to know.
It got pretty weird.
It gets pretty weird, yeah. So just warning you. Okay, so for example, I am likely tolerant of lactose. So I have the gene, that Northern European gene. I am actually predisposed to weigh more than the average person. Then you have things about I'm likely to weigh more on a high saturated fat. The thing that's really interesting is these haplotypes refer to your mitochondrial DNA. So my haplotype T, I think it was, or Y2B. Anyway, I can't hardly see that up there. Yeah, Y2B is, you can actually trace that back to a group. So my mitochondrial group originated right here.
And if you can have your mother, that's why we're doing it.
Right.
If your mother will actually do it and link, they can actually give you even more information.
Right.
Which is really stinking cool.
Right.
Yeah.
And then things like the Neanderthal ancestry. I think I'm slightly less Neanderthal than the average European.
Really cool.
So the interesting thing about the Neanderthals is that we thought that we didn't interbreed with them. There was this idea in the past when I was going to school that we just actually killed them all off, right? So we eliminated them, but it turns out that we did interbreed a little bit because we actually have Neanderthal variants in our genome that they can trace back to the Neanderthal fossils that they find. Okay. And then the ancestry part is really interesting. You can actually find relatives, cousins. I think I've only found like a third cousin on here. They write to me and I don't write back.
Because who needs more awareness?
Yeah.
Would you like that? All the same stuff. I'll share.
Okay, so this is like a little pie chart showing where I come from, right? So I am 99.9% European, and I don't understand, I don't know where this 0.1% Native American, East Asian came from. And then 0.1% was unassigned. So you can kind of find out where your ancestry comes from, where you come from in terms of your DNA. The other thing that they did is that this tells you if you're predisposed to being more sensitive or less sensitive to drugs. So for example, I found that in my genome, it says that people with my variation, maybe not necessarily a gene, but people with my variation, my nucleotide sequences are more sensitive to warfarin, which is a blood thinner, right? So I would need less warfarin than a typical person because of my genome. So obviously the concerns is that people will use this inappropriately, right? So like to determine whether or not you can get health insurance would be a really bad way to use it. And also you could, see maybe you would have to job for jobs. If they could have access to this, that might be a bad thing as well. So there's lots of confidentiality surrounding this stuff. So they do provide you with a measure. They tell you that this is going to be confidential and they're not going to share it with any other entity. But the big thing is that big database that we're creating, which is going to tell us a lot about the genome and the variation within it. Okay, so the way that this works is that you actually, they actually use what is called a DNA chip. So if we look at this genetic testing, right, this is what they use in the 23andMe, something very similar to this. And so what it is a chip, a piece of glass that has on it a strands of DNA. And there's single strands of DNA. So a DNA chip has, let's put DNA chip. Okay, so this has single strands of DNA.
Is that diagram in our book as well?
No. Okay. Single strands of DNA, and then a person's DNA, the test DNA, is added to the chip, and it is also single-stranded. So where complementary pairing happens, colors will change. Okay.
How old do you have to be to do this?
18, I think.
Really? You can have your children?
I think you have to be 18, but I'm not sure. Because it sent me a thing that said I could.
You could ask, no, the question is,
But
do
my
you
youngest
have the
child-
right to get your children's DNA tested? See, I had to apologize to my older child because I had them fingerprinted, like literally fingerprinted. And I'm like, and then I didn't my other one, and I was like, I'm sorry, but your fingerprints are in the database. So you have to behave. My younger one, I'm like, I thought second about that. You don't want to be fingerprinted. I'm not going to force you to be fingerprinted.
Here's your chance.
Here's your chance. Yeah, so the question is that violate their privacy? I'm not sure. It's a good question. So this just shows a chip, and yours would be unique, right? So where these lighted up, that is where your DNA found a match with the genome, the sample genome. And then this is actually just put into a computer. And the computer reads the variation in lights and where the lighting up happens. And then it spits out your report, which the 23andMe then translates into information. Okay. So one of these could be the sequence that tells them that I am lactose tolerant. One of these could be the sequence that tells them that I have a slightly higher predisposition for Alzheimer's than what is found in the normal population.
So if something doesn't light up.
Something doesn't light up, then that means that you have, well, maybe they all light up to some extent, but that means that you don't have that variant. So like if you were being tested for, they wanted to see if you had that variant for Alzheimer's, and like if this was it and it didn't light up, they would be like, oh, you don't have the variant for Alzheimer's, so you are at a lower risk of having Alzheimer's.
So would they tell you that, or would they only tell you the things that you have variant? Like that you're
no, so they have traits. So they have Alzheimer's and Parkinson's and maybe dementia, and I'm not sure if they do alcoholism. Depression, I think, is one. Depression. And so they have specific sequences that they're looking for. And if that sequence matches up, then they give you information about that. And as they find more traits, then they give you more information. So 23andMe is kind of nice because they continually update your profile and you only pay once. So that once they find out more information, they give you more information.
And your additional people, like, I think I paid $150. I think the original one was $150 for me. And then when my mom signed up this week, when she came to visit, she was cheaper.
Mm-hmm.
So each additional,
interesting.
your children, it gets cheaper the more people, because they want you, as you said, to build like a database, they want you to have multiple people, because that tells them something in itself. If you have, like, my sister, my mom, and my 2 daughters, and me will all be on there. So we can see, and then we'll be able to see a whole bunch more. But each person,
As long as you
gets
don't get
cheaper
bites.
for each person.
Okay.
Yeah, so
Yeah. So if you go to the doctor, you can also get genetic testing for specific diseases, right? And you can find out if you're a carrier as well. So I mentioned like Huntington's disease would be a disease that you could go in and you could be tested for. This is a disease panel specifically designed for people of Jewish ancestry, because there is one population in the Jewish population that has a higher incidence of particular diseases than others. So you could actually go in and have the doctor specifically test for these. So you can't rely solely on the direct to consumer. Like if you thought you specifically had a genetic disease or were a carrier for a genetic disease, then you would go in and you would be tested specifically for that. And so you still want to use the doctor route in particular instances. Okay, so we have lab today. Remember to bring your lab notebooks, but you're not going to turn them in, right? We're just going to turn in your lab notebook at the final exam, which is next Wednesday, not this one.
You want me just to bring my notebook Friday then?
Yes, I do.
Okay. All right, I'll do that.
And if I'm not there, you just put it in that tray outside my office.
Okay.
I've seen that tray, it's lab books.
Yeah. And, yeah, because like right after it, I'm leaving, so I don't know if I'm.
You could turn it in Thursday if you wanted to.
Okay, I'll probably do that
Okay.
then. And then do I get it ready? Like, I just go to the.
Yeah, did you contact them though? Because it's a small space now because of that little outbuilding. So you might want to call them and set up an appointment and say, can I come in at such and such time?
Okay.
Just to make sure they have a seat for you, because there's only like room for like 6 people.
Okay, yeah, I got to talk to him.
Okay,
So since you are my academic advisor, I
Uh-huh.
was planning on registering for summer term.
okay,
Where are we going?
Can I meet you sometime this week?
yes.
Sweet.
Can you come tomorrow? Or we could probably be able to do it right after lab. Lab is going to be really hectic though,
Okay,
and for me at least.
well, I can do it either Wednesday or Thursday, because I have to come up to town Thursday for an occupational therapy.
Do you know what you want to sign up for? Because you also need to
I
do
have
fall.
it on here. I'm not doing fall here.
Okay,
Yeah, I'm
well,
doing
just tell me
fall
what you
here.
want. Tell me what you're doing, and then I can just okay you to register. Because you probably don't want to wait, because summer classes fill up fast.
Yeah.
Where are you going to?
Fall term going to EOU.
Oh, nice.
Yeah.